Students, today we are going to talk about apoptosis. It may be also called as programmed cell death. So, what is the definition of apoptosis or programmed cell death? This is a normal occurrence which is happening to a cell in which an orderly sequence of events that lead to the death of that cell. So, if a cell having some sort of irreparable damages or maybe at the end of its lifespan, then a series of events may be occurring into that cell, and finally that may be lead to the death of that cell. So these dead cells may be recognized by phagocytes and finally engulfed. So what are the characteristics of apoptosis? The first sign or symbol of apoptosis is the shrinkage of the cell in volume, and the second thing is the loss of its adhesion from the neighboring cells, and the third thing is the formation of the blubbings or sutures on its surfaces, and then its contents may be leaked out of the cell, and then the next event is the fragmentization of its chromatin into small pieces. So finally these cell debris may be recognized by the macrophages, and those may be engulfed, and that's the end of the story. Around thousands of cells in a human body die due to apoptosis per day. The next question is, what sorts of cells are the victims of apoptosis? Say, if a normal cell, if it undergoes a series of mutations or DNA damages, normally the repair enzymes may try to recognize it and try to repair the damages. If the damages are irreparable, then these cells may be recognized or marked for apoptosis. And the second good example is that of neurons. You know that in normal cases the neurons may grow, and finally it finds its target tissues. Target tissues may be another neuron or some other cells. If the neuron fails to find its target tissues, then these neurons are also the victims of apoptosis. And the third example which I have noted here in this is the T lymphocytes. You know that if a body is exposed to an antigen, then a large population of T lymphocytes may be arised within the cell, and only some sort of or a limited number of T lymphocytes, those are producing specific antigens, can recognize that antigen, and those all other T lymphocytes may be eliminated from the body by means of apoptosis. Now we can look at the molecular mechanism which occurs during apoptosis. In the case of C. elegans, C. elegans are free-living transparent nematode, normally that used in research purposes. So in the case of C. elegans, Z3 is the major gene that plays a crucial role in apoptosis. So that is in the case of C. elegans, and in the case of mammals, instead of Z3, the caspases comes in role. So these caspases are the homologous proteins of Z3 in C. elegans. So what are the functions, or what are caspases? They are a group of cysteine proteases. Proteases means protein degrading enzymes. Cysteine proteases means they having these enzymes having cysteine at the key catalytic site. So these caspases are a group of cysteine proteases. They activated at an early stage of apoptosis, and then that triggers a series of events that finally led to the cell death. So these caspases that recognizes and degrades a large number of proteins, that involves protein kinases like focal adhesion kinases, protein kinase B, protein kinase C, and other proteins like RAF1, lamins. Lamins means nuclear lamins that keeps the intact shape of nucleus, and intermediate filaments, actin, tubulin, and gelsolin. In addition to all these, that activates another protein, that is CAD. CAD means caspase activated DNase. They are an example of endonucleases. Nucleases means there are two kinds. Nucleases means they are DNA degrading enzymes. They are of two kinds: endonucleases and exonucleases. Exonucleases that degrades the DNA from both ends, and endonucleases that degrades the DNA or cut the DNA or cleave the DNA internally. So this card is an example of endonucleases. So normally this card that may be attached with an inhibitory protein. If the caspase activates this CAD, then actually what happens is that this activation that cleaves the inhibitory protein that may be attached with the CAD. So the inhibitory protein may be get eliminated, and the CAD get activates, and then this activated CAD may be translocated from the cytoplasm to the nucleus, and within the nucleus this card activated card that degrades the DNA. Apoptosis that can be triggered by two kinds of stimulus. One is internal stimulus and the other one is external stimulus. Coming to the internal stimulus, actually the stimulus that is emanated within a cell. For example, the abnormalities or mutations those are developed or accumulated within a DNA, that is a sort of internal stimulus that actually activates an intrinsic pathway. So this internal stimulus that activates the intrinsic pathway. So in the case of external stimulus, the stimulus is actually coming from an external environment, like hormones or some other factors like cytokines, etc. See here in this example, if a body experiences a low level of testosterone, then actually that leads to the apoptosis of epithelial cells of the prostate gland. So here in this low level of testosterone, that is an external stimulus that actually leads the apoptosis of the epithelial cells of the prostate gland. Okay. So that's why you may heard that this prostate cancer that can be treated with the drugs that can interfere with the testosterone production. So these sorts of external stimuli that actually activates the pathway, that is extrinsic pathway. Now we can move on to the details of extrinsic pathway of apoptosis. As I have said before, in the case of extrinsic pathway of apoptosis, an external factor that comes in role. Examples of external factors are exposure to ionizing radiation, elevated temperature, viral infections, or experience in toxic chemical agents. So these are the external agents. These external agents that activate the immune system. Once it gets activated, then it secretes some of the factors like tumor necrosis factor or TNF. This is an example. That means this is a kind of a messenger. If it is a tumor necrosis factor, then it binds with its own receptor, that is TNFR receptor. Here it is TNFR1. Talking much about TNFR1, this is a protein that belongs to the family of protein having death receptors. This is a pre-assembled trimer. See, there are three subunits, pre-assembled trimers on the plasma membrane. It's having extracellular domain, transmembrane region, and a cytosolic domain region. This cytosolic domain region that may be composed of around 70 amino acids. This cytosolic domain that mediates the protein-protein interactions. See, if an external messenger or external factor that activates the immune system, then that activates the messenger. That messenger comes and binds to the external surface of this TNFR receptor. Then that leads a conformational change to the external surface of the or external domain of the receptor, and that conformational change that passes to the transmembrane region, and then it leads to or passes to the cytosolic domain region. And here in the cytosolic domain region, that leads the recruitment of another two proteins. They are procaspases 8. Coming back to the procaspases, they are the precursors of caspases. Actually, these caspases are associated with an inhibitory protein, but these procaspases having low proteolytic activity. Hence, if two procaspases associated together, then the proteolytic activity of each of these procaspases subunits that cleaves one of those inhibitory proteins associated, and hence there forms 4 polypeptide chains. They associate together to form a complex that is known as caspases. So this caspase doesn't have the inhibitory protein. That's how this is an active one. See here, these two subunits are procaspases 8, and the proteolytic activities of them that cleaves the inhibitory proteins, and get spliced out, and the final polypeptides associated to form the caspase 8. That caspase 8 activates another caspases and other protein kinases and other cards, etc., and finally that led to the apoptosis events.